Okay, so we're going to finish up this chapter by talking about allergic reactions, and then some of the issues related to transplants and rejections, the issues related to autoimmune disorders, and then finally we'll discuss a little bit about how the thymus changes over the course of the lifetime. So here we go. So allergic reactions are an immune response to something, to an allergen, which and these allergens, these are they're materials that otherwise wouldn't be harmful, but because they trigger a reaction in the body of a severe inflammatory response, then we see the allergy symptoms that we associate it with. So it may be nasal congestion as inflammation occurs in the nasal passages. It could be a full-on anaphylactic reaction as the entire body begins to respond to the allergen. Allergens, those are just another term for antigens that trigger allergic reactions. And when we look at allergic reactions, we can see that they can be classified into one of four types: immediate reaction allergies, antibody-dependent cytotoxic reaction allergies, immune complex reaction allergies, and delayed reaction allergies. And they are numbered type 1, type 2, type 3, and then IV means 4. So if you're not familiar with Roman numerals, this is the Roman numeral 1. It looks like a capital I, the Roman numeral 2, the Roman numeral 3, and then V is actually the Roman numeral 5, but if you put an I in front of the V, then that subtracts 1 from 5 to equal 4. Okay, so that's a brief introduction to Roman numerals. We're not going to worry too much about them. Okay, so let's look at the reaction types, and we'll start with the immediate reaction allergy, which is a type 1 allergy. This is the anaphylactic type of allergy, and it's a very rapid allergic reaction. It occurs or begins within minutes of exposure to an allergen, and usually that allergen is a food product of some sort or an insect sting. And it results in the overproduction of IgE, which then attaches to mast cells wherever those IgE antibodies are found, and to basophils. Remember, basophils are the white blood cells that release histamine and other chemicals. And so when IgE binds to mast cells and basophils, it releases histamine, another material called prostaglandin D2, and sometimes these leukotrienes that and they can sometimes cause extreme inflammation. And these immediate reaction allergies may lead to a condition called anaphylactic shock, and the signs and the symptoms for anaphylactic shock include itching, hives, so these eruptions across the skin, sort of raised, elevated, very itchy patches of skin, vomiting and diarrhea, and the primary method of treating an anaphylactic reaction is the administration of epinephrine. It's given intramuscularly, and so people who carry a device called an EpiPen, these EpiPens will deliver a pre-measured dose of epinephrine, and generally people who carry them are instructed to deliver it directly to their thigh muscle so that they can get it in quickly into the body. And sometimes because of the inflammatory response that's occurring in the airway, a tracheotomy may be required to ensure that the airway remains open. And a tracheotomy is an opening cut into the trachea in the cervical region in the neck, so that an open airway can be maintained. If a person has food-related allergies, it turns out that these 8 materials are responsible for about 97% of all food-related allergies. So eggs are a big thing. Everybody knows probably now. Peanuts and tree nuts both can cause allergic reactions, anaphylactic style reactions. Cow's milk and milk products. Soybeans are also very active. Fishes, including shellfish. Wheat and peas, interestingly, are all capable of initiating these kinds of allergic reactions. Okay, so I think that's everything I had to say about a type 1 reaction. Type 2 reactions, that's the antibody-dependent cytotoxic reaction allergy. And this is an allergic reaction to a foreign cell introduced in the body. And moderately paced, takes sometime between an hour to 3 hours to develop. And when this happens, antibodies bind to antigens on the cell membrane of this foreign cell, and they stimulate phagocytosis and complement-mediated lysis of antigen-bearing cells. And a blood reaction, mismatch reaction, is an example of this kind of allergic reaction, the antibody-dependent. And so, not a... Yeah, I don't have anything more to say about that, sorry. Let's see. The immune complex reaction allergy, a type 3 reaction. This occurs as large amounts of antigens wind up overwhelming the immune response, leading to, rather than having large clumps of antibody-antigen complement complexes forming, you wind up with really small clumps because there's so many there's so many antigens and so few antibodies, and so it makes these clumps of antibodies and antigens that are all agglutinated. It makes them really difficult for the phagocytes to find and remove because they are so small. And so again, this is a moderate rate reaction, sometime between 1 hour and 3 hours to fully develop. And it winds up that these small complexes that the phagocytes are having a hard time finding can block the smallest blood vessels, get into the capillaries, cause blockages because they turn into circulating emboluses. And so when you block off those capillary networks, then any of the tissues that are downstream of this blockage will be deprived of oxygen and nutrients, but oxygen is going to be what gets them first. Autoimmunity reactions are, and that is the inability to tolerate the self-antigens, are one example of a type 3 allergic reaction, because there are so many of the antigens in the body, because it's a self-antigen, then again, relatively small numbers of antibodies will cause tiny little agglutinations, not larger ones, and you can get this downstream blockage as the emboli lodge. Okay, so last type of allergic reaction is the delayed reaction allergy, and this is the result of generally of repeated skin exposure to chemicals. So household chemicals, industrial chemicals, sometimes even cosmetic materials that can stimulate this delayed reaction allergy. And it is a very slow progressing allergy. It can take up to 48 hours for it to fully develop. What happens is that the foreign material, whatever it is that's coming into contact with the skin, will activate T cells, which happen to be very abundant in the skin. And the T cells then, and as well as any macrophages that are attracted, release chemical factors that result in inflammation of the skin, dermatitis, the inflammation and eruptions of skin tissue. And so you get lots of uncomfortable, itching kind of, bumps that form all over the skin. May or may not be itching, I suppose. And so we talk about that as contact dermatitis when it's some material that we've come into contact with. And so a good example here might be an allergy to, say, poison ivy. So that toxin, the poison ivy toxin, gets on the skin, and then within a couple of days we wind up with really itchy, weepy sores that are caused, a result of this contact dermatitis. All right. So when we think about immunity, then it's also smart to think a little bit about tissue transplants and the rejection that can follow a graft or a transplant. So a tissue rejection reaction occurs as the recipient's immune function recognizes, if the whatever tissue has been transplanted, it recognizes the host. I'm sorry. Let's try this again. With the tissue rejection reaction, the person who has received the transplant, that person's body recognizes that this donated material is foreign, and so as a response, it attempts to destroy it. And so that can prevent successful transplant when the body engages in a tissue rejection reaction. And so the rejection reaction can be very great extent unless we can suppress the immune system with other drugs that will help to prevent that rejection reaction. Sometimes, though, now this is the opposite effect. If the donor tissue is immunologically active, then it may produce antibodies against the recipient's tissues, and that's called graft versus host disease. Is when the graft begins to fight the host, the recipient of the donated materials, and because the graft itself can produce immune cells. So in order to prevent either of these kinds of I hope I didn't destroy your eardrums there. So in order to prevent these kinds of graft versus host or tissue rejection reactions, then transplant recipients are frequently required to use immunosuppressive drugs. These are drugs that prevent or decrease the production of T cells and or the production of antibodies. But if you can do a good job of matching the donor to the recipient immunologically, then perhaps this immunosuppressive therapy may not be required. Okay, so what are the types of tissues that can be transplanted? If a person receives an isograft, iso means same, then that means that the transplanted tissue is taken from an identical twin, somebody with the same genetics as the person, but not the same individual themselves. Auto means self, so an autobiography is the story that I write myself. An autograft is when a bit of, say, skin or muscle or whatever is being taken from somewhere else in the recipient's body and moved to a new location. An allograft is when the transplant is taken from another person, the same species, but that person is not an identical twin. So you find a closely matched, immunologically matched donor, but someone who may be related but not all the way down to an identical twin level. And then last of all, xeno means completely other, I think is the term, xeno. I'm not sure of that now, but it is. It means that a different species is involved. So a xenograft is when we take, say, a heart valve from a pig heart and transplant it into a human, or maybe bone marrow or thymus tissue from a baboon and move it into a human. Those would be xenografts. So in the first three cases, these are all human-to-human graft. This is other animal-to-human graft. With an allograft, it's just another immunologically matched person. With an isograft, it's an identical twin. And an autograft is when the recipient's own tissue is used. Okay, so we're getting close to the end here. Let's look at autoimmune diseases. These are diseases that occur as the immune system produces what are termed auto-antibodies, so self-antibodies, and then cytotoxic T cells that will attack, that shouldn't say attach, it should say attack and damage and sometimes kill cells in body tissues and organs. And so there's a couple of examples that I've given you, and there's more even in the book. But autoimmune hemolytic anemia is when a person becomes anemic because autoantibodies are attacking and lysing the blood cells. Autoimmune ulcerative colitis, that's when the autoantibodies harm the colon. Itis means inflammation. Col is referring to the colon. So autoantibodies harm colon cells and leads to all sorts of intestinal pain and distress. Rheumatoid arthritis is another kind of autoimmune disease. In this case, it's the tissue of the joints that are being attacked. That means joints. Itis, again, is inflammation of the joints. Autoimmune that should be insulin dependent. Oh, daggone it. It's in your notes correctly, but that's the insulin dependent. Diabetes mellitus. And with insulin-dependent diabetes mellitus, it's insulin-dependent because you can administer insulin to the person to make them better. And the reason that insulin-dependent diabetes mellitus arises is because autoantibodies attack the cells, the pancreatic islet cells that produce the insulin. And then the fifth kind of autoimmune disease is systemic lupus erythematosus. So lupus is a really challenging disease to identify, and I don't believe that to date there are any particularly, like Hail Mary successful kind of treatments or therapies for lupus. But in lupus, autoantibodies attack the skin, the joints, the kidneys, the brain, many organs all at the same time. And so people who deal with lupus are really dealing with a lot of damage throughout the body and lots of discomfort. So where in the world does autoimmunity come from? Well, there's a couple of ways that a person can develop autoimmunity. The first is that it may be mediated by a virus. Part of how viruses work is when they inject their genetic material into the cell, it can there can be some merging between the virus DNA and the human DNA. And so sometimes as new virus particles are being built using the DNA that came with the virus as the template, then some of the human genes can be, or characteristics can be mixed into that, into the viral DNA. So once the virus is released out into the body fluids, the new copies of the virus that were made in the cell, then the immune cells learn the antigens on the virus surface, including some of these self-antigens. And so pretty soon we're making self-antigens, or antibodies against these self-antigens. So then the antibodies are going to go and attach to other normal body cells, and with the antibodies attached, signaling for the complement to come and join them. And then the complement there, you can see how it goes. We wind up attacking a lot of our own body tissues. So it can happen because of viruses and the way viruses accidentally incorporate human DNA into their own DNA. It can sometimes be that T cells just never learn to distinguish self-antigens from non-self antigens. And so, we just never get that ability. Or it could be that a non-self antigen, just by chance, resembles a self antigen. And in this case, I don't know how that got in there. Quality control. I need to get my editors to be a little bit more aware here, I guess. So if this non-self antigen happens to resemble a self antigen, then we start producing antibodies against the non-self antigen, and they may act against the self antigens as well. Okay, some more ways to develop autoimmunity. It could. I see what happened. Yep, I was supposed to remove both of these from this slide so that I could do it all here. So here's a whole story now about this non-self antigen that happens to coincidentally resemble a self antigen. Some ways that this can happen. There's a group A, Streptococcus bacteria, causes strep throat, and along the way can produce a larger body reaction or disease called rheumatic fever. And it turns out that this group A strep bacteria carries on its cell membrane an antigen that resembles the same as the antigen in a heart valve cells. And so people who have experienced rheumatic fever can wind up, through these autoimmune disorders, can wind up attacking the cells of the heart valve, which would be a bad thing to happen, no doubt. And then there's a really crazy phenomenon called microchimerism. A chimera, that's actually a Greek mythological creature, but it's an organism that's formed from multiple, the cells of multiple organisms. And so in Greek mythology, it's different animals that are all kind of clumped together to form one, parts of different animals that form one individual. So you get this really bizarre character. Well, this can happen with mothers, where the fetal cells make their way into the mother's circulation, and they wind up kind of setting up shop somewhere in the tissues, and they just kind of go inactive. They don't really make a big fuss, but they're just there. But then stress or other characteristics can really bring on the activity of these fetal cells, and as the fetal cells become active, then the body begins attacking the fetal cells. And along the way, because it's attacking the fetal cells, it also winds up attacking the tissues that the fetal cells have taken up lodging in. And so this idea that mother's body might have fetal cells, it's her own baby cells incorporated into the tissues of her body, can then lead to these autoimmune disorders where we start attacking the activated fetal cells and damage the tissues that they're living in as well. All right. So one last thing to talk about, and that is the condition of the thymus over the years of one's... Oh, no, almost done. Interesting. Okay, so on this one we've already talked about these. And so one specific issue that this microchimerism can cause is a disease called scleroderma. and I encourage you to go to the internet and look up some pictures of some folks who are dealing with scleroderma. It's where tissues harden inappropriately, and so sometimes it's even, it's like this, just affects one side of the body. Very unusual autoimmune disorder. And there may be, I think this is still, a piece that scientists are studying, trying to understand. But other autoimmune disorders may relate to this microchimerism. Okay, so last, just talk about some of the immunodeficiency diseases. And with immunodeficiency diseases, the body either never has or it loses the ability to fight off specific infections. And one way that this, one cause of immunodeficiency may be a genetic one, in which case it's named severe combined immunodeficiency disease. And with this severe combined immunodeficiency disease, from birth, the T and B cells may be defective and unable to do the job of fighting off any disease or infection. And so if you've heard or seen video of a young fellow that was named or kind of commonly referred to as the bubble boy, it's the boy who, when he was born without a functional immune system and so had to live in a clean environment, finally, unfortunately, got an infection. He was in his late teens, I think, and got an infection and it killed him. So it can occur genetically or it can occur because of disease. And so the human immunodeficiency virus, HIV, is the causative agency for AIDS, the acquired immune deficiency syndrome. HIV targets helper T cells, and it uses them to replicate and winds up killing them in the process. And without functional helper T cells, or any kind of helper T cells around, then the B cells cannot become activated and produce antibodies. And so while it's not the lack of T cells that kills a patient, instead it's the disease that can't be fought naturally within the body that winds up killing the infections, the patient. So viral infections or even minor infections, things that we can clear up very easily if we have functioning immune systems, can kill folks who are dealing with HIV and AIDS. Okay, last slide. It's a little bit about the thymus. The thymus reaches its maximum size during adolescence, so somewhere around 10 to 12 to 14 years of age. Thymus will be as large as it's ever going to be. By the time a person reaches age 70, the thymus will have shrunk by 90%, so that it's only 1/10 the size that it was at age 10 or so. And so as a result, with smaller thymus, or sometimes even a non-existent thymus, then we see a great reduction in the ability of the body to fight infection, because we're not producing as many T cells, and so we can't train and activate as many B cells. And so this 70-year-old is only going to have about a 25% as vigorous response to an infection as the 10-year-old might. And so for this reason, we see that older individuals are much more susceptible to the effects of diseases than our younger individuals. So there we are. Everything you ever wanted to know about immunodeficiency diseases and autoimmune disorders and the like. So happy studying. As always, if you are confused about something, do not hesitate to make contact with me. I'll be serving office hours both Monday and Tuesday. Monday and Tuesday. So feel free to join me, especially out there at the ARC. All right, study well.